Answering MCAT questions in under 60 seconds. A scientist is investigating whether fatty acid breakdown is functioning normally in muscle cells under fasting conditions. To verify that beta oxidation is proceeding efficiently, which of the following is the most appropriate metabolic indicator to measure, right? Let's look at these answer choices. So A, the accumulation of pyruvate in the cytosol, right? Well, pyruvate is a product of glycolysis, not fatty acid oxidation. So that doesn't make sense to measure in this case. B, the import of long-chain fatty acids into the endoplasmic reticulum. Well, this would be part of the process of fatty acid synthesis and subsequent modification, right? So again, that wouldn't be appropriate to measure if we're trying to detect fatty acid breakdown, which is oxidation. And then C, the generation of NADH and FADH2 during mitochondrial fatty acid catabolism, right? Well, that sounds right because fatty acid catabolism is fatty acid oxidation, and NADH and FADH2 are the primary reducing equivalents that are formed in that process and deliver electrons to the electron transport chain so you can drive ATP synthesis. So that would be an appropriate thing to measure here. And then D, the conversion of acetyl-CoA into citrate in the cytoplasm. Well, this conversion of acetyl-CoA to citrate happens for the purpose of fatty acid synthesis, not fatty acid breakdown. So again, that would be inappropriate to measure, and therefore C is the best answer.